Here we start with an MCQ about a lipid bilayer. As you all know, lipid bilayer, it forms core of all biological membrane. We are going to see this MCQ. There are 4 options which have been given. Please have a glance at them. We are going to discuss them one by one. Symmetric nature, exchange of phospholipids, and then some permeability characteristics. Symmetrical nature. Is the membrane symmetrical? There are 2 leaflets, upper and the lower. This is phospholipid bilayer precisely. And outer leaflet and the inner leaflet. Let us see like that. So this is cytoplasmic and this is exogenous. This is the external one. So this figure explains certain things. For example, oligosaccharide chains are attached to certain proteins to form glycoproteins. Some oligosaccharides are attached to lipid portion of the membrane to form glycolipids. And these are present they face towards the oligosaccharides. They face towards the extracellular space. So that makes the outer leaflet as different from the inner leaflet because the outer leaflet contains mostly the oligosaccharides, the glycolipids and the glycoproteins, so as to say. Secondly, as you can see, the proteins are also asymmetrically distributed. So oligosaccharide portions of glycoproteins and glycolipids face the extracellular space. And then second point, asymmetric orientation of protein. The third point which establishes asymmetric nature of the membrane protein is that lipid distribution is also asymmetrical. For example, the outer leaflet contains most of the phosphatidylcholine and lecithin. Likewise, some other phospholipids like phosphatidylserine and serine and cephalin, they are present in the inner leaflet. So that really proves that the nature of the membrane is not symmetric. The first option ruled out. Second, is there rapid exchange of phospholipids between 2 leaflets of the bilayer? Leaflets are shown in a magnified view in this figure, outer and inner. What this question means is it possible for this particular phospholipid to move from this outer leaflet to the inner leaflet? Is it possible? So this we are going to examine. This is the polar portion of the phospholipid. It forms interaction with the surrounding water and with the neighboring head groups also, with the neighboring polar portion of the neighboring phospholipids. All these interactions have to be broken if it were to move. Is it possible? Not an easy thing. So this is this moment called flip-flop moment or the transfer moment. There are 2 requirements. The polar head group has to abandon its interaction with water, electrostatic bond hote hain, and also with the neighboring head groups. Not an easy thing to do, very much unfavorable. And secondly, it has to dive across the hydrophobic barrier. What do we mean? जैसे ये polar group है and membrane lipid is mostly, it is hydrophobic because of these non-polar hydrocarbon chains of the fatty acids. So we call it hydrophobic core or more appropriately hydrophobic barrier. और पोलर पोर्शन का यहां से डाइव अक्रॉस करना, पूरा मूव करना, that is least likely or rather impossible. So this option also, rapid exchange of phospholipids between two leaflets, this also ruled out. I must add one thing, that lateral movement and rotatory movement as depicted in the figure are possible because there are no such constraints on these movements. No bonds are to be broken. Third option, lack of permeability for steroid hormones and small fatty acids. We simply have to go back to that earlier video and recall that membrane is predominantly hydrophobic. So small hydrophobic substances can pass through it. So steroid hormones and small fatty acids, because of their predominant hydrophobic character, both can move across the hydrophobic barrier easily. Recall that the steroid hormones, they enter the cell and elicit their action. Their mechanism of action is like that only, that they enter the cell. Why? Because of their hydrophobic nature, they are able to easily cross the hydrophobic barrier. By the same logic, polar substances cannot cross. So third option also ruled out, lack of permeability, because it is permeable, so rule out. Fourth option, impermeability for small inorganic ions. We have named two, potassium and chloride, because these two are polar. And among polar also, if you see permeability coefficient, जो charged substances होते हैं, their movement is maximally restricted across the hydrophobic barrier. So impermeability, yes, impermeable. So this fourth option is the correct option.